Hello, and welcome to the unit on gene expression. Um, what this chapter is all about is how do our segments of DNA or our genes actually make us, or how does the gene of a bacteria actually make the bacteria? How do our genes keep us alive, and how do they function? This is going to involve turning off genes when we don't want to use them, turning on genes when we do. It's going to involve how do our cells, every one of our cells, for the most part, contain all of our DNA. The DNA that makes our eyeball, the DNA that makes the stomach enzymes, which help digest food, they're all in every cell. How do the cells of the stomach know to make those enzymes? And how do the cells of our eyes know to make a lens or know to make an iris or the parts of our eye? That's what this chapter is going to be about. But not only us, it's going to be about all organisms. So we're going to start with prokaryotic gene expression. And this is how do genes in prokaryotic cells, bacteria, make certain genes do certain things. I'm not going to go everything in these PowerPoints. You guys can read them, and make sure you always read these speaker notes down here at the bottom. That's going to be important to your understanding of each slide. But this first slide talks about two things in general, and I'm going to try to explain those really quickly. If we want to make a protein called tryptophan, or an amino acid called tryptophan, then we have a gene, our prokaryotics cells have genes that can actually make that. Now, there's times when they want to make it, essentially if there's none present, but there's also times that they want to make it, if there's none present, they're going to want to make it. If there is some present, they're not going to want to make it. So you can do two things. You can create an enzyme that can regulate this enzyme activity. These enzymes are all made from these genes on this DNA. This DNA is making genes that eventually lead to the tryptophan. Or we're making mRNA strands that would then make, proteins that would essentially make the tryptophan. And they can create something that can mess with these functions of these enzymes so they don't actually get all the way to the tryptophan. Or they can do something that can regulate these genes. We're going to be all through this lesson talking a little bit about both. But either way, they can stop the DNA from making the final product. This is some more information about eukaryotic cells versus prokaryotic cells. This is where it all starts, and that's with the operons. An operon, as you can read, is a group of genes, all grouped up next to each other, usually on a strand of DNA. And they all kind of have related function. There's usually a promoter area of the DNA where RNA polymerase can bind onto and make an RNA strand that can go off and make the gene, or can go off and make the protein. And that's what an operon is. It's a segment of genes, all related. We're going to be talking about these operons as we go through. There's a couple parts of an operon that you need to know. You have a promoter region, which is where the RNA polymerase can bind on and begin to make the RNA strand. You actually have an operator region, which is a segment of DNA that an inhibitor can bind onto. A repressor protein can bind onto and actually inhibit the transcription. So if something's bound there, you can't make the rest of this gene, essentially turning the gene off. There's also a regulatory gene, usually somewhere further up the DNA strand, that can make an mRNA that can then go make a protein. That's a repressor protein, and that's the actual protein that can bind to the operator. Remember, we can bind right here, shutting off the rest of the gene, essentially turning it off. And that's called a regulatory gene, and it's somewhere further up. It's what actually makes this active repressor here. But then we have modulators, and these things can affect the repressor. We can have this modulator here, which can turn this repressor off, which will not allow it. If it's shaped like that, it can't fit in here. That's a modulator. I'm going to stop the video and pick back up again with a new video. Ready for that one?